my name is Don Green, and this is my 6.3 FRQ assignment. The RNA polymerase enzyme is a large complex made up of multiple subunits. The prokaryotic form of RNA polymerase has 4 subunits capable of transcribing all types of RNA. In eukaryotes, these enzymes have 8 or more subunits that facilitate that attachment and processing of DNA throughout transcription. 1. Initiation. Initiation begins when RNA polymerase wraps around the promoter region of DNA. The promoter is a DNA sequence that guides RNA polymerase on where to bind upstream of a gene. While prokaryotic RNA polymerase can directly bind to DNA promoter sequences, eukaryotic forms require the assistance of transcription factors for initial binding. Once RNA polymerase successfully binds DNA at the targeted promoter region, the enzyme can continue with the second stage of transcription. 2. Elongation. Elongation commences when RNA polymerase unwinds double-stranded DNA into 2 single strands. These DNA strands are used as genetic templates for RNA synthesis. As the DNA template strand moves through the RNA polymerase, it builds an RNA strand that is complementary to the transcribed DNA strand. 3. Termination. Termination is the final step of transcription. Once RNA polymerase encounters a terminated sequence or signal, it stops adding complementary nucleotides to the RNA strand. This is followed by the release of the RNA transcript, which marks the end of transcription for that template of DNA. A spliceosome is a large ribonucleoprotein complex found primarily within the nucleus of eukaryotic cells. The spliceosome is assembled from small nuclear RNAs, or snRNA, and numerous proteins. Small nuclear RNA molecules bind to specific proteins to form a small nuclear ribonucleoprotein complex, which in turn combines with other snRNPs to form a large ribonucleoprotein complex called a spliceosome. The spliceosome removes introns from a transcribed pre-mRNA, a type of primary transcript. This process is generally referred to as splicing. A codon is a trinucleotide sequence of DNA or an RNA that corresponds to a specific amino acid or stop signal during protein synthesis. DNA and RNA molecules are written in a language of 4 nucleotides. Meanwhile, the language of proteins includes 20 amino acids. Codons provide the key that allows these 2 languages to be translated into each other. Each codon corresponds to a single amino acid or stop signal, and the full set of codons is called the genetic code. Genetic code includes 64 possible permutations or combinations of 3-letter nucleotide sequences that can be made from the 4 nucleotides. Of the 64 codons, 61 represent amino acids and 3 are stop signals. For example, the codon CAG represents the amino acid glutamine, and TAA is a stop codon. The genetic code is described as degenerate or redundant because a single amino acid may be coded by more than 1 codon. When codons are read from the nucleotide sequence, they are read in succession and do not overlap with one another. The ribosome is a complex cellular machine. It is largely made up of specialized RNA known as ribosomal RNA, as well as dozens of distinct proteins. The ribosomal proteins and rRNAs are arranged into 2 distinct ribosomal pieces of different sizes, known generally as the large and small subunit of the ribosome. Ribosomes consist of 2 subunits that fit together and work as one to translate the mRNA into a polypeptide chain during protein synthesis. Because they are formed from 2 subunits of non-equal size, they are slightly longer in the axis than in diameter. Ribosomes are minute particles consisting of RNA and associated proteins that function to synthesize proteins. Proteins are needed for millicellular functions, such as repairing damage or directing chemical processes. Ribosomes can be found floating within the cytoplasm or attached to the endoplasmic reticulum. Their main function is to convert genetic code into an amino acid sequence and to build protein polymers from amino acid monomers. The structure of tRNA can be decomposed into its primary structure, its secondary structure, and its tertiary structure. The cloverleaf structure becomes a 3D L-shaped structure through coaxial stacking of the helices, which is a common RNA tertiary structure motif. The lengths of each arm, as well as the loop diameter in a tRNA molecule, vary from species to species. The tRNA structure consists of the following: a 5' terminal phosphate group, the acceptor stem, the CCA tail, the anticodon arm, and the T arm. While the specific nucleotide sequence of an mRNA specifies which amino acids are incorporated into the protein product of the gene from which the mRNA is transcribed, the whole of tRNA is to specify which sequence from the genetic code corresponds to which amino acid. The mRNA encodes the protein as a series of contiguous codons, each of which is recognized by a particular tRNA. One end of the tRNA matches the genetic code in a 3-nucleotide sequence called the anticodon. The anticodon forms 3 complementary base pairs with a codon and mRNA during protein biosynthesis. That is the end of my 6.3 FRQ.